Do a video about an organization called Openlongevity, and I'm going to start with an introduction by a former Facebook friend of mine before I was deplatformed, of course. She's a wonderful young lady, and I want her to explain what Openlongevity is about, and then I'm going to show you a little bit about the website and maybe some interviews with her.
There's one problem we usually face in life extension community and life extension field. A weird ask from potential sponsors, potential volunteers, from people who are being introduced to the ideas just first time. They usually ask for, better proof that it's possible. That it's actually possible to extend human lifespan, especially dramatically, especially crossing, natural threshold, and preferably on human, of course. Yeah, I understand that, but let's take a look at it from a different perspective. Imagine a person drowning, just he or she just fell into the water, asking for help, looking, at us on the beach, and we're like, yeah, sure, we'd like to save you, of course, but maybe show us some proof first. Prove that it's possible to save you. Like, what? We don't do that. We don't do that in our lives, right? Because we value the human life. When the question is about, life extension technologies and anti-aging, for some reason we're asking for some proof here. We don't need to prove that it's possible. We need to prove otherwise, that it is impossible to save humans. It is impossible to extend human lifespan. And when or if we do that, yeah, we probably might stop. But unless we do that, we need to try. You just need to understand what you value here. And if it's human life or your personal life you value most, then you need to try. Easy as that. You don't need any other proof. You just need to understand what your values are.
So this is the Openlongevity website, and we're going to navigate this and see what's going on here. Okay, let's see. So, let's see. We're going to go about open genes. Okay, Openlongevity. We are extremely lucky to be alive. Life can be even better, and it can last longer. We are against aging and all causes of death, and we support using scientific methods to fight them. How big is the community? Team members, over 8,600. Community members with different social media, 27. Volunteers, 13. Aging is complicated. Aging is complicated, but we need to figure it out during our lifetime. We are turning to science for solutions: molecular biology, computer science, chemistry, sociology, and interdisciplinary research. We're focusing on the boldest pioneering solutions, often going where others refuse to tread. We will never give up. We consider the task of finding a cure for old age the most important one. It is a scientific task, and the majority of our effort is being applied in this field. But we have to be honest with ourselves. The research and development in anti-aging lack the needed scale and pace. That's why we are also in constant search for better solutions, innovating on every level to support the development of new approaches to old ones. Anastasia Igorova, co-founder and CEO. These projects are not just our projects potentially. These are your projects too, is what they're trying to explain. Okay, so this is collecting information on geroprotectors. Let's take a look at the geroprotectors. Is that how that works with that? Okay. This is a database designed to help researchers study genetics and aging and related processes. They manually collect information on the evolution of the gene, its functions and methods to determine if the gene is associated with aging and life expectancy. The idea of Immortality Buyers Club is to conduct open experiments that would help us get closer to radical life extension of the human lifespan. We can believe the selection of combinations for gene therapy to be the most promising direction. Using the developments of the cutting-edge mathematical physics, meaning they're going to study the aging of various organisms, analyzing it as the dynamics of a complex network system. Their mission is to develop therapy that prevents age-related degradation of the extracellular matrix. The extracellular matrix is what your cells live in. It's kind of like the medium that keep your cells together. Okay, they are focused on small molecules that have potential to slow down aging and protect people from diabetes, cardiovascular disease, Alzheimer's disease by inhibiting cross-linking. Remember, I brought up cross-linking a lot in the extracellular matrix. See, what happens is when you get cross-linking, you get stiffening. And when tissues stiffen, they don't work the way they're supposed to. And that's where you get diseases like heart attacks, strokes, all sorts of issues. Since biological aging is a complex phenomenon, this document discusses the widespread possible range of fundamental research and applied developments in the field of aging and body rejuvenation. All research. The reason I'm not going to the read more is because I want people to understand they can obviously go to this website and read these things on their own. Okay, all research and development proposed by this roadmap should make it easier to achieve the goal of reversal the biological aging of a person. The biological aging is not separated from biology and medicine in general. And that's what I want people to understand. Transhumanism, or life extension, I like referring to it as life extension, but transhumanism is, in and of itself, the same thing as medicine. Okay, there are many startups and large pharmaceutical companies that develop drugs directly or indirectly related to aging mechanisms. They do not always state it openly, but this doesn't make them less interesting, rather the opposite. In Openlongevity, we believe in the power of art, so we present you with Expedition for Immortality, Misha Batin's first philosophical movie, blah, blah, blah. We need to figure out the best strategy for all people and companies involved in the industry, preferably the United Strategy. Here are the tasks for our volunteers. That's in Russian. Okay, let's take a look here. This is the Open Genes. This is what the databases look like, so you can search the databases if you're a researcher. So let's take a look here. It's loading. So here you go. This is the Open Genes database. The Immortality Buyers Club. Let me get out of here. Aging is, okay, this goes on about that. Okay, this is, okay, I wasn't going to read more. This shows you to read more, okay. So, this is about transhumanism, vision. Okay, let's go into the research roadmap. Goals of this roadmap. The major goal is to define the search for ways to stop and reverse biological aging. Thus, the roadmap's goal is to increase the scale of scientific research in the field. Our document is ready for implementation in grant policies and public and private funding programs. It can become the basis of mega collaborations like the Human Genome Project. By solving the fundamental problems outlined in the roadmap, we can expect a cumulative increase in human life expectancy and health span. The roadmap shows the areas of research, okay? Molecular mechanisms of aging and rejuvenation, aging of individual tissues, organs, and body systems, and the search for mechanisms for their rejuvenation, population studies on aging and rejuvenation, systemic approach databases and mathematical models, prevention of age-related diseases, which are the main cause of human death, instrumental and technological methods of rejuvenation and preservation of life, the formation of clinical practice in the fight against aging, meaning getting this stuff into doctors' offices. Molecular mechanisms of aging and rejuvenation. Okay, so this is some of the problems: destabilization of the genome cells, reduction of chromosome telomeres and the end sections, that's like the ends of the shoelaces. Okay, the emergence of aging-associated epigenetic modifications of chromatin, violations of proteostasis, mitochondrial dysfunction, an increase in the number of senescent cells, meaning what happens is cells that don't divide anymore, they become senescent and they release bad signals to other cells to mess up their functioning. Loss of cell sensitivity to nutrients, again, that's something they need to reverse. Stem cell depletion, how can we fix that? Well, with stem cell therapy, of course. Aging of the extracellular matrix, occurrence of protein cross-links, and impaired intercellular signaling. So as you can see, aging it's not one thing, and that's what's important to understand. Aging is a multifaceted thing. The roadmap offers research of aging mechanisms and the development of drugs that block the molecular mechanisms of aging and promote rejuvenation of the body. Cell genome destabilization. Fundamental research in this area includes studies of the mechanisms of age-dependent changes of reparation system, defects in nucleus architecture, amplification of transposon mutagenesis. I used to be able to say mutagenesis with a lot more ease. The effects of somatic mutagenesis on cells, meaning that the cells themselves mutate. High-capacity methods of assessing the intensity of somatic mutagenesis and the identification of cells with damaged genomes, the influence of environmental factors on destabilization of the genome of the cells, identifying molecular mechanisms of increased stability on the genome in long-lived species, including naked mole rat, Brandt's bats, blue whales, and other animals, the therapeutic methods of selective activation of individual reparation pathways, for example, induction of repair by homologous recombination, methods for removing repair scars expressed in the activation of repair systems in the absence of DNA, clinical research, the development of gene therapy approaches to the treatment of diseases caused by genome destabilization and malfunction of repair system, in particular progeria, which is a horrible disease that causes premature aging, which shows you, let me put this out here, by helping to fix the aging problem, you are helping to fix most diseases, okay? Most diseases are based on the senescence, premature senescence of a body system or another. Werner syndrome, Bloom syndrome, xeroderma pigmentosa. Okay, my scientific lingo is a little bit off. I haven't been socializing with my science friends lately. Clinical trials for gene therapy of cancer and also therapies which perform preventative removal of cells with mutations in oncogenes. The development of approaches to therapy for age-dependent destabilization of the genome, in particular the inducible increase in the activity of DNA repair systems, and the development of approaches that prevent the accumulation of DNA damage in vivo, meaning in the body, in vitro, which arise from the divisions of cells, the development of molecules that are radioprotectors and anti-mutagenic agents, meaning they protect you from radiation and they will stop you from mutating. Expected outcome: understanding the contribution of mutagenesis and specific mutations to the aging process and creating tools for leveling dangerous mutations, non-toxic drugs that activate repair processes and eliminate cells with dangerous genome damage. Reduction of chromosome telomeres. So fundamental and clinical, identifying the real contribution of telomere reduction in human aging. It's iffy whether telomeres actually play a role in that, but it seems to. So there's good evidence both ways, but it seems like there's other evidence that says not really that much. Okay, ways of reactivating telomerase activity in cells of various tissues and organs. A comparative study of the terminal sections of chromosomes in individuals of various species and phenotypes, and identifying the causes of accelerated telomere contraction. The role of telomeres in the formation of pathologies and disease. The identification of diseases caused by telomere reduction. The study of clinical consequences of telomerase gene therapy in humans, meaning does it mess things up if you activate this gene? Possible approaches to the treatment of telomere reduction, pharmacological and gene therapeutic methods of reactivating telomerase in vivo, meaning in the body, transplanting cells with elongated or more protected telomeric sites. Expected outcome: telomerase reactivation therapy in dividing cells to the level of telomere length that does not affect the aging process. The onset of aging-associated epigenetic chromatin modifications, the dynamics of DNA epigenetic modifications, occurrence and histones in different types of cells and tissues at different ages and in normal and pathological conditions, the relationship between these modifications and the functional state of cells, tissues, and organs, identification of the pathological epigenetic patterns and pathways associated with the restoration of the chromatin epigenetic state, associated with youth and health in general, the role of sirtuins and other histone-modifying enzymes in longevity, the influence of factors affecting the change in DNA methylation, chromatin architecture, synthesis of non-coding RNA, and changes in transcription patterns for longevity, the age-related dynamics of transcriptome, changes in different tissues and organs, the transcriptome profile stabilization, the epigenetic hours of aging. So this is how are we going to count the epigenetic age? How do we really know how old somebody is, in particular at the level of different organs? So again, if you read Dr. Morgan Levine's book, True Age, she talks about how some people's immune systems age faster than their cardiovascular system. Some people's cardiovascular systems age faster than their pancreases. Some people's pancreases age faster than their brains. Some people's brains age faster. You get what I'm saying? So there's different epigenetic stages, and a lot of diseases such as diabetes could be related to the epigenetic aging of the pancreas. Clinical studies of the role of epigenetic modifications of chromatin in the formation of pathologies and age-related diseases, study of the contribution of epigenetic factors in oncogenic, meaning basically in cancer development, the development of epigenetic therapies for aging. Expected outcome: understanding the contribution of epigenetics to overall aging phenotype, targeted therapies correcting epigenetic alterations, proteostasis disorder, fundamental research in this are the dynamics and changes of the cells, the proteome, in various tissues during aging, and the case of age-related pathologies, the relationship of proteomic changes, meaning changes in the cells, the functional state of cells, tissues, and organs, the role of proteotoxicity factors in aging, including heat shock, endoplasmic stress, and oxidative stress, the pathways of regulation of activation, autophagy, proteasomal degradation, chaperone-mediated protection and protein refolding, the role of protein aggregates in aging, as well as the pathways of induced degradation of aggregated proteins, the role of activation of stress tolerance, factors including FOXO transcription factors. I'm going to get into that when I talk about phytonutrients and how to activate the FOXO the FOXO gene. The search for pharmacological inducers of autophagy and proteasomal degradation of damaged proteins, the state of proteome in patients with various age-related diseases, expected outcome, proteome stabilization and stress reliance therapy, mitochondrial dysfunction. As you can tell, they are trying to hit everything that causes aging with research so that they can then so what they're trying to do is get as many people together to share information from as many fields as possible so that we can finally conquer aging, and with aging, all of these other diseases. So mitochondrial dysfunction, research in this area, age-dependent disorders that occur in mitochondria in the cells of different types of different tissues. The role of free radicals and antioxidant systems in maintaining cell homeostasis, cell damage, and cell aging. Relationship between stress reactive oxygen species, and cell proliferation, mitochondrial dysfunctions that are not associated with free radicals in particular, the destabilization of the mitochondrial genome, the disturbance of the relationship between mitochondria and other cell organelles, mitochondrial-induced inflammation, permeabilization of mitochondrial membrane during stress, impaired mitochondrial bioenergetics, mitochondrial division disorders, mitochondrial dysregulation, and calcium signaling, induced apoptosis, the balance of fusion and fragmentation of mitochondria, and various... See, mitochondria are interesting. They're literally a bacteria that merged and lives in symbiosis with the human cell. It's very interesting, but they're the powerhouse of the cell. The search for ways to rejuvenate mitochondria, leading to an increase in life expectancy, specifically with regards to the activation of mitophagy, mitohormesis, and so mitophagy would be the eating the mitochondria, and mitohormesis would be using the stress adaptation. How can we stress the mitochondria so that it can adapt? The separation of the process of oxidative and phosphorylation in mitochondria, the development of gene therapy for mitochondria, in particular aimed at stimulating mitochondriogenesis through activation of PGC-1 gene, approaches to mitochondrial rejuvenation, ex vivo growth of mitochondria, delivery of mitochondria to cells, and mitochondrial transplantation, the contribution of mitochondria to the formation of pathologies and age-related diseases, expected outcome, a therapy that renews mitochondria and restores their functions, an increase in the number of senescent cells. Okay, so we have to find a way to get rid of them. Fundamental research. The mechanisms of senescent cells formation, for example, cell cycle arrest, genome damage, activation of tumor suppression genes, INK4A, and P53. Now, I want to be sure that everybody understands this. I am not a scientist. I am not researching these things. I simply know of these scientists. Many of these scientists I was friends with before I was deplatformed from Facebook for my political views. But I want to make sure people understand that. So, if I mispronounce things, I'm sorry. The search for new markers of senescence and previous states of senescence, the contribution of senescent cells and proteins secreted by them. Remember I said senescent cells, they no longer divide, but they secrete these signaling chemicals that corrupt non-senescent cells. Characterization of senescent-associated secretory phenotype in different tissues, meaning how do these cells differ? How do they look different and act different in different tissues? The relationship between senescence and cancer and impaired immune function, tissue and organ specificity of senescence, meaning, again, how does this change from organ to organ? The search for ways to destroy senescent cells with senolytic therapies. Senolytic therapies essentially just get rid of the senescent cells, which makes the person more youthful. Quercetin was thought to be a senolytic. It may or may not be. I think the jury's still out on that one. The development of therapeutic agents that return stem and differentiated cells from senescence to normal state. Clinical studies of the role of senolytic therapies in preventing pathologies and increasing life expectancy. Identifying diseases and pathologies caused by senescent cells. Studying gene therapy methods of cleansing the body of senescent cells. Expected outcome. The therapy allows targeted and tissue-specific destruction or rejuvenation of senescent cells. Loss of cell sensitivity to nutrients. Fundamental research in this area. The role of catabolic and anabolic signaling in aging and longevity. Age-related dynamics of the activity of the somatotropic axis, growth hormone, insulin-like growth factor, insulin, and intracellular processes in response to these hormones in different tissues in humans and different ages. The disturbances in molecular pathways, recognition of nutrients as a factor in aging and inductor of age-related diseases, the mechanisms of increase in life expectancy. that increase in life expectancy, the contribution of intracellular factors involved in the, so these are the different metabolic pathways, mTOR, AMPK, and NAD+, as targets for potential therapies of aging and age-related diseases. Clinical studies of therapies that suppress the way insulin-like growth factor and insulin signaling, in particular the study of rapamycin and metformin to prevent age-related pathologies, clinical trials, okay. Stem cell depletion, okay. I'm going to get back into this. This will be part 1. And then I'm going to start again at stem cell depletion. I think I'm going to cover all of this because this is quite interesting and I want my viewers to see this stuff.